Okay. So this is GIDAS week 3, and we're going to be going over GEO. So I think let's start by just recapping everything from last week, or the past couple of weeks actually. So for example, this is how you can see your data set off of GEO or like NCBI, and you can go to this link and you can search up, for example, this GSE ID. And it'll take you to the data set, which is this one. And you can see that this data set is a good data set because it has control groups, which is the normal samples, and then it also has experimental groups, which are the tumor samples. So that's how you can look at a data set if you have the GSE ID. And so another thing to note is that the organism is like the species that was like taken, where the data was taken from in the experiment. And the type is the type of data that was collected. So in this case, it was expression profiling by array, which is like RNA data. And when we're looking for data sets for schizophrenia, we're only going to be looking at data sets that have like this RNA data. And the series is the ID number that is used to locate this data set. Kind of similar to the GSE ID. So we also went over microarrays, and microarrays are a laboratory tool used to detect the expressions of thousands of genes at the same time. And so DNA molecules are attached to each side to act as the probes to detect the DNA expression. And then the unknown DNA molecules are cut into fragments, and then fluorescent markers are attached to these fragments, and then they are allowed to react with the probes of the DNA clip. So here is an example of a data set. I think this is the same one that we searched up before. And so again, just like the requirements, I guess, for choosing a data set, it should usually have 30 plus samples, so you know that it's as accurate as possible. And the sample should have a control group and a disease group. And so Grace and I were looking through the data sets that everybody put in for their homework for last week. And it looks like some people didn't have a control and a disease group. And so basically when you're looking at the data set, you should look at the sample section right here. And you should see if there's like two different groups. Like here you can see that there's a normal group and there's a tumor group. And so that's why this data set would be okay to use. But for example, if there was just one type of data that was just like used as samples, then that would not be a good data set. And then also a significant disease, a significant difference should be seen between the control and the disease group. And so we'll be going over that this week. Yeah, when we analyze the data set. So here is GEO2R. Wait, Grace, is this the one? Oh, never mind. Wait, is this the data set that we were talking about, the bipolar one?
Yeah.
Okay. So this is, so basically, Grace and I were looking through the data sets yesterday for schizophrenia, and we spent like quite a bit of time looking through data sets, but it looked like none of them really had like good correlation between the control and the experiment group, and there was like not a significant difference. And so that's why we decided to look at this bipolar data set because it also fell under mental health. And so here is the data set that we will be using for research this year, I think most likely. And so here are a bit of just like vocabulary. So when you look at upregulated and downregulated genes, which we'll see in GEO2R when we analyze the data, an upregulated gene is a gene that is more active than normal, and a downregulated gene is a gene that is less active than normal. And you can also call them overexpressed or underexpressed genes. And so another thing is the log FC value, which is like log fold change. And this is also shown through the graphs in GEO2R. And this is basically a statistical value that indicates whether a gene is over or underexpressed in disease samples compared to the control samples by like positive or negative and by how much, which is like the absolute value of the number. And if you have a larger log fold change value, it indicates a more significant result. And then another important idea is the p-value. And this is a measure of how likely it is that the statistical results occurred by random chance. So basically, you're taking a look at whether there's actually a difference between the experimental group or in the control group, and you're basically trying to see that it's not just based off of like random chance that the results have occurred the way they have. And so a small p-value indicates that the difference between the two statistical groups is most likely just due to like random occurrence. And the cutoff p-value for looking at this is 0.05. So if the p-value is greater than 0.05, the data is significant, I think. Wait, did we write this wrong, Grace, on the slides?
No, it's less than 0.05. It's significant.
Yeah, okay. Okay.
Oh.
And then if the p-value is greater than 0.05, then the data is not significant. Okay, so do you want to talk about this?
Okay. So for this data set, since our schizophrenia ones didn't have great correlation, we just decided to use the one that I'm doing with my group in MiraCore, which was about bipolar disorder. So the data set we chose was the postmortem brain samples, and it had a large demographic, and their results were they grouped the people into bipolar positive and then the healthy control. And then there were significantly a great number of upregulated and downregulated genes. So then, Vidi, should I do the demonstration?
Yeah, sure. Go ahead. I'll stop sharing.
I can't share.
Oh, you can. Let me give you access. Okay, I'll just make you a host. I'll just make you the host, okay?
Okay.
Okay.
Here we go. Can you see it?
Yeah.
Vidi?
Yeah, we can see it.
So if you have this data set from like, just search it up in Google, and then you go to the NCBI website, you go down to analyze with GO2R. So then you can analyze like the specific like cases. Like for this person, you can see all their like data and their disease status, and then like all this other information. And so we want to sort it by like the bipolar and healthy control. Like down here, you could see that there's a lot of healthy control. So then you want to sort them first with disease status, and you want to define your groups with control and bipolar. So to select the data, you just like click it and then use to select all of them. This might take a while. So then after, you will be able to see like the upregulated, downregulated genes, and then like the volcano plots. Vidi, did you go over that?
No, I got over the volcano plots. Let's just analyze this data and we can talk about it.
That is a control. Okay, usually it's a bit faster because there's not as much information. Like this one has the age and then like the years of illness and then everything else. But it's usually a lot shorter. Oops. And then that'll be the control. And then we analyze with GO2R. And hopefully it'll work.
So basically what Grace did is she took like the data and like for each patient and she split it up into control and disease groups. And then when you click analyze, it'll like give you graphs that like go over like the differences between the control and the experimental group. So yeah, you can see here. And then this is where like the p-value and stuff like
Yeah.
starts getting.
So first we can see like the visualization. So like in here, the fold change, as you can see, like they're comparing the genes, and the larger like the fold change, there's a greater like up regulation or down regulation. The blue is the down regulation and the red is up regulation. And usually there'll also be distinction in the UMAP, but there's not much in this one. So then here you can see like the p-values. It sorts with the lowest p-values, and then like once you get lower, you'll see that they get larger. And then they also have the log FC, which is the fold change. So if you click on this, you can see like this one isn't that great, but maybe this one. You can see that the control has a higher, the genes in the control are more upregulated compared to the bipolar. So then that may indicate that like this gene, the SUCLA2 gene, might be a factor that causes the bipolar disorder. And then you can also see like, since this one is negative, this means that the gene in the bipolar disorder is more upregulated, and the control, the gene in the control is more downregulated. Okay. Should I go over the downloading the table?
Yeah. Should I share the slides, or if you have
Yeah.
the slides up, you can just like show them really quickly?
Oh yeah. Okay. So here in the GO2R, you can download the table for this. It's the button like right here.
So
And
if you
then-
guys are following along, you can download the full table. You want them to download the full table, or should we just show them?
I already have a full table in the meeting.
Okay.
Yeah. So then the full table should look like this. It has like the same thing as the table from GO2R. And where to go? Okay. So next we are going to be analyzing the data with Stringdb, which is like seeing the connections between the protein encoding genes. So then we can determine like what factor the genes has in the bipolar disorder. So then first, step one, as you can see down here, I labeled, it says the downloaded table. So this is it not modified. As you can see, there's a lot of them. And then to make sure your data is significant enough that there's like correlation between the two groups, then you want to see the p-value. So in the second spreadsheet, I cut off the numbers that were below 0.05 to see that like these were all the ones that are significant enough. And then the third one was the gene selection. So I used this function to basically get all of these gene symbols that have the NM in front of the gen bank accession. And the NM before the gen bank means that the genes are protein encoding genes, and those are the ones that we want to be looking at in Stringdb. Okay. Should I put this in Stringdb and show them, or you're muted?
Yeah, I think we can go over like the basics of Stringdb, because it looks like we have enough time.
Okay.
And then, how about at this part, does anybody have any questions? Because I know we're moving kind of fast. So does anybody have any questions?
I'm good, thank you.
Okay. Shreya, are you good?
Yeah, I'm fine.
Okay. Okay, so we can move on then.
Okay. So I'm gonna just select all the genes over here and copy and then paste them here. There's like a lot of them. And I selected about like the first, wait, humans, or humans. Okay. Let me see how many there. There were 357, and that's a lot. You can do to like lessen the load. You can just do like 100 because right now it's very busy. So maybe just try 100 and then it'll be easier to look at. So in here, you can see like all the colors and then all the genes that are a part of our bipolar research. And then you can see, where's the key? The legend. Okay. So you can see from like the colored lines, there's you can see the different like correlations between the different types of genes. So we don't really look at the color from, we don't need to look at the color from this, but you can see that like the more connected it is, the more like the more connections it has. Does that make sense? I don't even know. Vidi?
Yeah, that makes sense. So basically, Stringdb is like showing you like the interactions between like different proteins. So that's what it is mainly.
Yeah.
And yeah, the more connections it has, the more connected it is. Or like the more strings, I guess it has, the more connected it is to other proteins.
Let me see. Maybe I can like delete some of it because it's
I think one thing you can do is, I remember you can also get rid of the ones that aren't connected to anything. You remember how to do that? Let's go back.
I'll have to look it up.
Wait, could you go, wait, could you give me control of your screen really quickly?
How do I do that?
I think, okay, here, I requested remote control.
Okay.
Okay. I don't know if I can scroll down. Let me see.
Oh shoot. Wait, let me do this.
Settings, and then I think it was, yeah, could you scroll down really quickly? I forgot what part. Oh, I
Oh,
think it's hide wait, go down. Hide disconnected nodes in the network. That one. And then, yeah, I think, yeah, now update it.
oh yeah.
Yeah, okay. So that looks less intimidating, I
Oh
think.
yeah. Okay. So you can see there's like a cluster over here. So then the genes over here are most likely have something to do with bipolar. And you can see like the COPS genes. Like COPS3, 4, 2, they're most likely to have something to do with it. And like the genes that are here are also seen like in the input. Oops. And then like here you can see a basic summary of what the gene does. So to analyze the genes, you can see from the analysis, there's like biological processes, like I am not going to try to pronounce that. And there's like various, what do you call this? Like biological processes that come with these genes that may or may not be like inhibited because of it, since it if it is like upregulated or downregulated. So like for this, if you search up proteasome core complex and like bipolar, there may be something that has a correlation with the proteasome core complex and how it affects bipolar disorder. And like, since a lot of these are the proteasome, maybe that has a connection and you could like research on that.
Yeah, so basically when we're doing research, we're trying to look at some of the connections in some of like the processes that are listed here. And we can like go deeper into them and like how they cause bipolar disorder. We can also look at how bipolar disorder may be connected to other diseases through like some of these processes.
Yeah,
There's like various ways to like do research based on these. So this is like very important.
yeah, like in KEGG pathways, it's basically you can see like how where the gene is in the cell and then how like it affects the pathways in your body. And as seen here, like the proteasome is pretty important in this process. And additionally, like maybe it has a connection to Parkinson's disease. So then, yeah. Oh, also, if you like highlight it, you can see like as seen here, Parkinson's is highlighted green, and then you can see all the connections, the green, those are the genes that have like connections to like maybe Parkinson's disease. And then you can see here, like there's a cluster of blues that are all like interconnected. And then that means it has something to do with the proteasome. And then like you can do that for any of these. Like in the signalsome, I'm probably butchering these pronunciations, but you can see like all of these, like these 4 genes are pretty interconnected, and it has something to do with this specific process. And then maybe you can do like more research into that on how it affects bipolar and just what it does in general.
So basically one thing I think we can do for like homework maybe is just look at some of these processes and then like figure out which one we want to do research on. I think a good one might be like the relationship between bipolar and Parkinson's. So I feel like that's like easier to explain to everybody. If we go into like maybe a very detailed process, it might be harder. But yeah, even there's like other diseases as well, like
Yeah.
recently getting one of them.
Like they show the genes to you in like bunches. So maybe since like there's like 2 clusters, you don't have to do all of the genes because that would be a lot. So like for this one, I don't know what that is, but you don't exactly have to do that one since there's all like so many of it here that you can focus on. Like the PSMA genes, there's like so many of those. So maybe you can just focus on that. And also when you like click on the genes in the like web, you can see the function, and then maybe you can see what it does. So then if it has like recenters network on this node, maybe this is probably not right, but maybe it has some connection to like neural networks, and then it has some effect on that aspect of bipolar disorder. And additionally, since we have the specific genes here, you could also go to gene cards and then do more research on that. Like, let me go to gene cards. Okay, let me choose one. PSMA2. I'm gonna do that. So you will see like this gene is a part of the proteasome too. So you could do research on the proteasome and like write about how exactly this gene has an effect on the proteasome, which has an effect on bipolar. And like this, oh, you could also do like this disease is also associated with cystic fibrosis, or maybe you could do another gene that's more directly connected to bipolar, or you could like connect cystic fibrosis to bipolar. But basically there's a lot of different like angles you can come at it from.
And then we're also going to be using other tools to help us with this research. So right now we're just trying to find like maybe a couple of processes or diseases that we can connect to bipolar disorder using like what we have in Stringdb.
Oh, shoot, these here.
Oh, okay. Well,
Um,
it's fine. Okay, just let her in. Okay, anyways, we have like 4 minutes left on this meeting. Do you want
Okay.
me to like reopen it? Should I do that?
Hi, Shruti. You missed the most important part.
Oh no, I'm sorry. I never got the notification on Discord for some reason. So I didn't even know it was 3 to 4.
Okay.
Okay,
Sorry.
it's okay.
Oh, we have 3
So-
minutes.
Yeah, we have like 3 minutes left in this meeting. Do you want me to, I'll end this one and I'll start a new one. Is that okay? Like, I'll just restart this meeting again.
Oh,
So
sure.
everybody just log off and then just log back on again.
Okay.
I had a
Oh.
question.
Yeah, go ahead.
Wait, so when you're looking at those data sets, are you looking at like you're looking at one disease that's there and like the genes that certain people have that are overexpressed when you're looking at like the GO2R, right?
Yeah.
Mhm.
So then you take those genes and then you like look at the ones that are protein encoded, right? And you put
Yeah.
those into the Stringdb, right?
Yeah.
Okay, I got it. And then from there, like whichever ones are protein encoded, you can like look at what they're related to, and then from there you can see if that's like related to the disease in the first place, right?
Mhm.
Okay, got it.
You can look at that more. I think also, Grace, do you want to send like a link to like the data table for like everybody to
Yeah,
look at it?
I will.
So like everybody can put it into StringDB now, and then we can help them like have StringDB set up. Because I feel like
When is the homework due? Since I don't know if we're having a meeting this week.
whenever the next meeting is, it's not really homework. It's just like we'll just do it in class. We'll just set up
Okay.
StringDB.
I
And
think
is there anything? Sorry,
you can go.
anything else that we're gonna cover that is important, or can I leave because I should go?
Oh, I don't think there's anything else, right now, Grace.
Mm. Yeah.
Okay, cool. I'll copy and paste the minutes of the meeting onto the Discord, so like whoever missed it.
Okay.
And if there's anything else, just let me know. Thanks, guys.
Thank you.
Bye.
Bye. Okay, Grace, do you want to send the link to the data table in the chat?
Okay.
The spreadsheet, I mean, not the data table. So we can maybe help Shruti and Shreya get like StringDB set up.
Oh shoot. Bro.
Me.
Wait, I'm a little confused. So are we looking at like certain diseases that we don't know what genes they're connected to?
So right now our disease is bipolar disorder.
Mhm.
So if you look at the genes, some of them, or like look at the processes at the bottom of StringDB, some of them show like connections to different diseases. If you saw like Parkinson's and like the other disease. And so what we're trying to do is we'll click on like, for example, Parkinson's disease, and on the StringDB map, like it'll have like a certain color, like blue, and then you'll see like the certain genes that are common between bipolar disorder and Parkinson's disorder. And so you can make conclusions based on that, like maybe something in like the genes is causing bipolar disorder and Parkinson's. Maybe you can say that Parkinson's patients have a greater chance of having bipolar disorder. So things like that, and then we'll just look into that more.
Okay, that makes sense.
Yeah. And I think Grace put in the data table in the chat.
Should I like demo it again?
Yeah, wait, do you guys want to share your screen so that we can like help you get to StringDB and then put in the data so you guys can see what we were looking at, if that's okay? Or if you want, we can demo it again.
I can demo it again.
Okay.
Oh, I need sharing.
Oh, okay. Let me make you host. Oh, okay.
Mm. Okay. Okay, can you see it?
Yeah.
So this is the spreadsheet from GO2. How do I move this thing? This is so
And this is the
okay. I don't know how to use computers.
So this is the data table that's in chat. So maybe you guys can open this data table really quickly. I think
Yeah, they're on.
Yeah, I don't know if
Someone's on.
yeah. So if you guys are on here, can you just do a thumbs up in reactions if you're on this? Okay. Okay, cool. Okay, so they're both on the data table. So now
Okay, I will explain it again. So we got this data table from our GO2R after we analyzed comparing the control group and the bipolar disorder group. And we got all these genes that you can see the data of that may be downregulated or upregulated, depending. I don't know where I was going with that sentence. Okay, we'll go back to the spreadsheet. So we have all of the genes that are significant in our research, which are the protein encoding genes. And we got this from the GenBank accession where it started with the NM, which means that they're protein encoding. And then we will copy this, all of that into Stringdb.
Okay, so can you guys just copy? You're looking at the first 250, right, Grace?
Something around that number.
Okay, it's fine as
Probably
long as it's
300.
like the first 300.
I do not know.
Okay, it's fine. So just copy whatever you're
Open
copying.
the Stringdb website, and
It's in chat.
yeah, so many tabs. I don't like tabs.
It's just like
I'm just talking to myself right now. Okay. You select the J column, and I assume all of you know how to do that. And then you copy it, and then you paste it.
okay, and then also when you go onto oh, never mind, you sent them the exact link. Okay, it's fine.
Yes,
And then for organism, put Homo sapiens.
yes, because we're humans.
Because the people that were in the data set are
Oh
human.
yeah, because they're humans.
So are you guys okay? Yeah. Now click continue when you get to this page. And you should see and then continue.
Ignore that.
Yeah.
I hope your computer doesn't crash.
Yeah. And then you guys should come to this page. So are you guys at this page? Can you guys do a reaction again? Like
Yeah, I'm on it.
okay.
Okay.
Shreya, are you good? Okay, Shreya's
Does
good.
yours look like this, or are there like colorful lines? Hello.
All right.
Mine is just like a list.
Mine is like colorful lines too.
Colorful lines?
Yeah, it's got the colorful lines on it.
Okay, good.
Wait, Shruti, wait, what's showing up for you?
It's just like a list of everything.
Can you press continue?
Yeah,
Oh,
yeah, yeah. Okay. Yeah, it should load and then you should see something. Do you see it?
yeah.
Okay.
Yeah, I see it.
Okay. So basically what Grace and I were talking about is that these are like different genes and like proteins that are important in bipolar disorder. And so the strings or like the lines you see between them are the connections that they have to other genes and proteins. And so we're mainly trying to look at ones that have like a greater connection to the other like proteins and genes like around them. And so the more like lines it has connecting it to other like proteins, the more connected it is. So like where Grace's like cursor is right now, that's like an area where a bunch of like proteins are like very much connected. So that would be like, those would be good proteins to look at. And then when you click on any of them, it gives you like a description of like what it is. So you can read through those.
What it does.
And so when you scroll down on, oh yeah, you can go ahead and,
Analysis?
Wait, how did you get the protein encoded genes on the side of the table, like away from everything else?
Oh, I used the spreadsheet function.
Oh, okay.
yeah, we just did it for you guys, but
Yeah. I'm not a spreadsheet pro, and I just Googled it.
Yeah.
Or I asked my mom, because she likes spreadsheets. Okay, you didn't even know that. Okay.
I didn't see it on the table though that like, was in the chat.
you don't see what? The gene selection column?
No, I don't see. Yeah, the gene selection column.
Oh,
Oh.
it's because you have to go to step 3 on the bottom.
Yeah.
See, like there's tabs on the bottom of the spreadsheet and it says,
Here.
you see it?
Yeah, I got it. Thank
Okay,
you.
and then just copy that column and paste it into the Stringdb link that we set in chat, and then just tell us when you get there. So I don't want to go, if we go too fast, it gets kind of confusing. So.
Oh, wrong one. Where'd it go?
It's on the very end. Yeah. And then, so Shreya, what step are you on? Are you?
I got the map now.
Okay, cool. And then you can scroll down
Oh.
and you can go to settings, settings.
So-
And then you can go scroll down and you can click hide disconnected nodes in the network. So check that box, the one that Grace is on. It's in the settings tab and it's at the very end in the advanced settings section.
Yeah, I did that.
Okay, cool. And then you can click update. And it should be like a much cleaner map because we don't want those other proteins and genes that are not connected to anything else. They're not significant to research. So now this one looks better. And so also, didn't you want to, you have to click evidence for the line to be
Evidence.
like colored.
Yes.
So Shruti, did you get the evidence part? 'Cause I think you were gone at that part.
Yeah, I got that.
Okay, cool. So do both of your maps look like this, like similar?
Mm-hmm.
Okay, cool. And then we can go to analysis. And here you can see the processes, the different types of processes. And then Grace, do you want to like explain this part again?
Okay. So the processes, as you can see here, it determines which genes are like significant in the process. So like for protein, I'm not gonna pronounce that. You can see like the highlighted red ones are the genes that are important in these biological processes, since these genes are also genes that are significant in the bipolar disorder. This protein, whatever, in bipolar disorder probably have a connection. And we could choose like a topic from any one of these, like biological processes, cellular components, and like KEGG pathways, any of these to do research on and like find a connection between like the proteasome and bipolar, or maybe like Parkinson's and bipolar, since they have similar genes that play a role in it. And then you can like highlight this, and then there will be like blue genes, and you can see they're like all over here. And yeah.
And so one thing I was saying is like, when I was talking about the diseases, because this is, I think, similar to the research that I did, you can look at, for example, Parkinson's, and you can like find like, if you click on Parkinson's and you see like all the blue proteins and like genes that are colored, you can kind of research those genes and see how they play a part in Parkinson's and how they play a part in bipolar disorder. And then you can kind of compare them and see like maybe why that gene is causing both, or how it's playing a part in both. And then you can make like a conclusion, like a bipolar disorder, like Parkinson's patients might have a greater chance of being bipolar and stuff like that. And you can also see like Alzheimer's disease and like Huntington disease. So there are like many different diseases that we can connect with bipolar. So that's also like one path for like doing research that we could do this year. And then there's also just like other processes.
Yeah.
Like I think Grace went over like the COP9 one. And so like
the specific genes you see when we're doing research, you can also search them up on gene cards. Like for the PSMA gene, as I said before, it's important in the proteasome. And then maybe if we wanted to connect that, we could also do research using this website.
Wait, Grace, could you send the link for gene cards in the chat so they can like look at it?
Okay.
Okay, cool. So that's gene cards if you guys want to like search up specific genes or like proteins for next week. And then one thing we were just telling, I think Duty asked about the homework. There's no homework, but just like if you want to get ahead or if you want to like look at stuff, you guys both have strings set up and you have like all of like the processes and like the diseases listed. So you can like look at certain genes or processes, you can click on them and kind of do a little bit of like preliminary research before we actually start. So yeah, if you want to do that, you can do that for next week.
Yeah, just know how to use it,
Yeah.
and then we'll go off on that like in the next weeks.
And then wait, Grace, could you go to the slides?
Yeah.
And then the last slide is the attendance.
I don't think we need an attendance form, right? Because we already know who came.
It just makes it easier, it just puts it on the spread.
Okay.
So just fill out
well-
the attendance form.
Please do this because Vidi says so.
Okay.
Let's see. Oh, I don't need to open it. What's the code word? Why
The
is
code
Duty's
word-
name on here?
Just put like, I don't know. How to the code word is
Is potatoes.
potatoes. Yeah.
Yeah.
Like obsessed with potatoes. I feel like our code words have just been things to do with potatoes now at this point. Okay, yeah.
Okay. Should I go over quickly Dr. Lee's announcements with
Yeah,
like the
sure, you can do
contest?
that really quickly. Yeah.
Okay. We have 9 minutes. I will talk very fast.
Okay.
Okay, so we have the 2022 Genes and Health Contest. So this is in February. It's February. They spelled February wrong. Did they spell February? That's funny. Okay. This is the Genes and Health Contest. So basically, the competition is basically what we are learning currently with like research. So like here, there's a multiple choice test, and then computational research, and then random short presentations. So basically, like presentations about our disease of focus, I think. Is that right, Vidi?
Yeah, I think, yeah.
Okay.
Oh, no, wait. Are you talking about the random short presentation?
Yeah.
Those are just like, so each GIDAS branch can make like a random presentation on anything. I think the last year's presentation that won was like Zoom etiquette for dummies. So you can just like Zoom etiquette for dummies.
Okay.
So it's
Yeah.
like a random presentation that's like supposed to be fun. It's for like bonding within like our branch. So yeah, if we like figure out research in the next like week or so, we can start working on that too. So that's just for like GIDAS branches. But like the top parts are, wait, could you scroll down really quickly? The main parts are the multiple choice test, and then the computational research. There's also an advanced contest and a general contest. I think we can sign up all of our members for the general contest, because we haven't gone over R yet.
Mhm.
And so yeah, R programming is something that we'll do like a little bit later. And yeah, this is like
Oh, right.
What
If
are like the
online-
multiple choice? Sorry, Grace, you can go.
Wait, you can go. I will figure it out
Oh,
later.
okay. For the multiple choice, is it on like our disease of focus?
no, so the multiple choice is on general, I think on like biology, actually. It's kind of
Oh.
like mRNA, RNA. It's about like just everything. And then it's also like like bioinformatics stuff. And oh, so it says right here, this year's disease of focus is like mental illness, but we're like focused, the focus is supposed to be depression, but we chose like schizophrenia and then we switched it to bipolar disorder. So just, I think it's generally on mental illness with like focus on depression, and then it's on like basic genomics and bioinformatics. And then it's also on vaccine information, which Grace and I actually haven't gone over. We should actually, like the MVA stuff. But it's basically
Oh.
like convincing other people to get vaccinated. There's a different program, and that's what it's about. So there's stuff on that. It's very much common sense based, but genomics and bioinformatics, and then mental illness is just like the two main categories on like
Yeah. And it is February 5th. So we have a month And
Yeah.
they just announced it like when? Like a week ago. Like
They just,
last
nobody
Wednesday.
knew the date. Yeah,
Yeah.
nobody knew the date and they just announced it.
So
And
we-
yeah.
if you want to participate, we can give you resources. Like they have resources that we can share with you, and we could also, since we haven't gone over like the very basics, like the UCSB genome browser, because I think there's gonna be something on that, so we can also teach a little
So
bit.
we can send you the study materials if you want to sign up, just let us know. And we can also sign you up. And then it's 3 hours, but like some parts of it will be like Zoom presentations for other GIDAS meetings. So some parts is just like optional. Like the actual contest won't be that long. And then there's also like awards for people that place.
You can
Uh,
perform at a talent show.
yeah, if you have a talent, you can perform at the talent show.
Yeah.
And yeah, it's a kind of like a fun event just with like but for biology and just for the GIDAS, like all of the GIDAS branches to kind of come together. So and it's also completely online, so you don't have to go anywhere.
Yes.
Okay, that's
And
cool.
you can like wear your pajamas or something.
Yeah.
Okay. Okay. I have 3 minutes and I need to go. So this is the 2022 Computational Biology Research Camp, and this is by MIRCORS, which is the like overarching umbrella umbrella
Organization.
organization for GIDAS. So they have like sessions and it's not that expensive, which like for other camps they're like thousands of dollars, which is this is pretty good. And so they have different topics like I think they have like R stuff. I don't know where it is, but you
It's
can
here. Just like this is basically what you'll go over, see the camp overview part. And so if you want to sign up for the camp, you can sign up for the camp. And then if you guys want to enroll in the genes and health contest that we just talked about, just like let us know. And this can all be found on the Miracor website where like Grace just sent the link to the summer camps. You can like find the health contest and other material in there. And if you want to join the contest, just let us know. We'll give you like some study material. And yeah, we'll also just let you know like the Zoom link and times and stuff. So yeah, I think that's it
But-
for
highly recommend.
you what?
Okay, we are good.
Okay. I think the recording is good too.
Yes.
Okay. So I'll send, I think Diti's gonna send like a quick summary and then I'll put the recordings on the Google Drive and I'll send you guys a link. And yeah, I don't think there's any homework. You guys can just come to the next meeting. If you guys can look at Stringdb a little bit more if you want to, but there's no like actual homework.
Yeah, I might still share my screen.
Okay.
Are the meetings in the future going to be still in person, or are we going to move to online?
I feel like I don't know. We're still kind of like in the middle.
I think
I
it'll
feel like
depend.
it depends, yeah.
Sometimes on Saturdays, a lot of people are busy because they have other stuff going on.
So the thing is,
maybe we can do some online and some in person.
it depends. I think our online meetings are more productive, but our in-person meetings have like way more people show up. So yeah, just depends. We'll think about it and then we'll let you guys know.
Okay.
So what do you guys think?
Yeah.
Okay, I feel like
personally
maybe
prefer the
this time a
online.
lot of people didn't come just because it was like the first meeting. But like if you make like a concrete time every Saturday, people will probably like free up time and then
Yeah, I
be able to
agree.
come
And I
down.
feel like this like online worked better for me versus like in person. Especially because in person aren't the meetings during lunch.
Yeah, they're doing that.
So like we have a lot less time, we're more constricted. And plus because people need to eat too, it's like I feel like it's kind of a distracted time.
Yeah. So online for sure works better. I think even I feel like these meetings are like way more productive because we're not stressed about time.
Yeah.
But I think we might
It's just
be
more
able
about
to-
attendance. Like if no one's gonna come to these online meetings, it doesn't like matter if we're more productive if there's less of us.
and we also have we can record the meetings too. So yeah, we'll think about it. Most likely we'll keep it online. We might change the timing if that works better for some people, so we'll see.
Yeah,
yeah.
we could do
Send
a
out-
poll and like ask if like
Yeah,
whatever time works.
I can do another one is good thingy, but for like the whole week.
yeah, you can do it for the whole branch, like everybody, you can put in announcements.
Yeah. Wait, did I not put it in announcements the first time?
No, you put it in leadership.
Oops.
Okay,
Sorry.
no, it's fine. Okay. Thank you guys for coming.
Okay. Bye.
Thank you. I'm so sorry for being late.
It's okay.
So nice.
What?
Your pictures in the back are so aesthetic.
Thank you. I worked very hard on them. And that clock's broken. Completely broken. It says 6 o'clock or something. It's not 6 o'clock.
Okay.
Okay, bye.
Okay. Bye.
Bye.